really glad to have all of you here today and kicking off this week of innovations in Parkinson's disease, a week of insights with UC San Diego experts. I want to thank you for attending today, and I really want to appreciate all of you who have been partners to our department for so long and supporting our center, supporting our Parkinson's Disease Center, as well as our department. I'm Dr. James Brewer. I'm a neurologist and I'm chair of the Department of Neurosciences at UC San Diego. We're extraordinarily proud of this department and its Parkinson's program as really the #1 NIH-funded neurosciences department in the nation. And I wanted to tell you just briefly about some of the special sauce that makes this place so key in innovating in care of Parkinson's disease and other neurological disorders. This center is housed in a remarkable multifaceted campus, and that is UC San Diego, which has not only a medical center, but also a school of pharmacy, a school of public health, and a remarkable mesa that includes top-notch engineering. So today's talk is going to be about how those centers come together to really innovate the care of Parkinson's disease. This week is going to be extraordinarily exciting for all of us because we're going to highlight to you what we've done in Parkinson's disease. Today's going to be about wearable technology, transforming symptom management. Tomorrow will be care beyond the clinic, community support programs. Wednesday will be unlocking LRRK2, a new drug target, which is going to be quite exciting and some real important work taking place here at UC San Diego about that genetic risk factor for Parkinson's disease. Thursday will be 4D biology and cell dynamics, paving the way for personalized medicine. And then Friday will be creating new neurons, the potential to reverse Parkinson's disease. So I think I'm going to be here with you this whole week, and I'm really excited to see what we're going to be highlighting with you. And I'm just delighted to have you here. So today I wanted to welcome you to talk about Parkinson's disease and wearable technology. This is where the Center for Parkinson's Disease has partnered with the Center for Engineering and Center for Wearable Sensors as part of the Institute of Engineering in Medicine, and really has come to innovate the wearable technologies as an approach to taking care of patients with Parkinson's disease. So we have two fantastic leaders, many of which you are already aware of. Dr. Irene Litvan, who is the Center Director of the Parkinson's and Other Movement Disorders Center, which is really a major feather in the cap of UC San Diego as a recognized center of excellence in many national institutes that have recognized the work that Irene Litvan has done. Irene is a board-certified neurologist, providing gold standard care for neurology patients, especially in patients with movement disorders. She's overseeing this program that has really brought national attention to our center. We are seen as a center of excellence not only by the Parkinson's Foundation, the Lewy Body Dementia Association, the Cure Progressive Supranuclear Palsy, and the Huntington's Disease Society of America. This really shows that this center is at the highest achievement of any of the centers in the nation. So Dr. Litvan will be joined by Dr. Joe Wang, who's a distinguished professor of the Department of Nanoengineering. So he has pioneered the development of nanomotors and nanoactuators, bioelectronics and biosensors, wearable sensor systems, and flexible materials. So the two of them have really brought together their expertise to apply it towards Parkinson's disease. And I'm really excited to hear what they have to say. So I'm going to welcome Dr. Irene Litvan here to kick off today's meeting. Thanks so much.
Hi, good morning. Thank you so much, Dr. Brewer, for your kind introduction. It is my pleasure today to present you a brief overview of our research, particularly on wearables. Although I'm going to be giving you the concepts and why we are using wearables, Professor Wang is going to give you all the details, so you'll understand much more than what I'm going to be saying, hopefully. So Over the past years, I've been studying early and accurate ways of diagnosing all the Parkinsonian disorders. In fact, that is what has led me to get an NIH Merit Award, as well as an Academy of Neurology Research Award in movement disorders. I am extremely interested in this because without having a good diagnosis, we cannot really understand the causes of the diseases or treat them appropriately. So my research has been focused on this, and obviously it is the major focus, understanding as well how we can measure all these problems that people have. Before going into the details of the presentation, I want to just remind you that Parkinson's disease has 4 stages. One that is the preclinical, in which only people that do have genetic factors and have an abnormal imaging can be diagnosed, and we're searching for markers for the diagnosis at an earlier stage. Then there is a prodromal stage in which the symptoms that occur are only non-motor. So there could be decreased smell, enacting dreams, constipation, depression. And then comes the early Parkinson's disease stage, in which slowness, stiffness, and resting tremor may occur. And then as the disease progresses, there is an advanced stage in which there could be major fluctuations, changes between the on and off and the dyskinesias, as well as presence of cognitive problems, including dementia, as well as orthostatic hypotension as part of the autonomic symptoms. So we do research in all these stages, diagnosing, understanding causes, treating the symptoms, avoiding the symptoms to develop, as well as searching for ways to treat the complications. So all the neurodegenerative diseases are proteinopathies. That means that there is a protein that deposits into the brain, and that is what leads to an abnormality in the configuration of these proteins that leads to the death of the cells. And that is what a neurodegenerative disease pretty much is. So this allows us to search for those abnormal proteins and search for biomarkers, as well as novel ways or strategies to treat these disorders. In the case of Parkinson's disease, this is alpha-synuclein. So we do have several studies that do look at the genes, as well as the causes of the disease and the search for biomarkers in not only Parkinson's disease, but also diseases that are atypical, non-Parkinson's disease, such as progressive supranuclear palsy, corticobasal degeneration, and dementia with Lewy bodies. So we want to understand the causes not only by studying genes, but also trying to look at what are the environmental exposures so we can prevent and find appropriate symptoms. So here, what we want to do is to discuss briefly the therapeutic studies that we have. Those are in clinical development, and those are phase 1, phase 2, as well as phase 3. Phase 1 means that we're searching for safety and also the right dose. Phase 2 is when we look at that particular dose compared to placebo, so we can see if the medication is effective and also safe. And a phase 3 is when the same is done in a bigger population, and that is what allows FDA to approve drugs. So the studies that we do have currently are the studies for Parkinson's disease using small molecules that make the proteins not to be changing configuration, therefore avoiding the aggregation. And we have another one that is avoiding the spread by using antibodies against alpha-synuclein. We also have studies in progressive supranuclear palsy, in multiple system atrophy, using antisense oligonucleotides, as well as nerve growth factors. So in addition to those studies that try to slow disease progression, we also have studies that do help the symptoms. And particularly, we have an interesting study using subcutaneous levodopa with a pump. And we compare that with oral levodopa and try to see if motor fluctuations are better treated than otherwise. In addition to that, we have a study for people that have low blood pressure, and that is with a medication that's called ampreloxetine. So now let me talk about wearables. That is what the whole symposium is about. And as you can see, the United States is really moving forward, and particularly our university, in view of our phenomenal bioengineer group, including principally by Professor Wang. So you can see how even that has changed regarding the market and how much there is an influence of medical devices and how much this market is going to grow as the year that do pass. So we use wearables not only for what was there, that is the management and the therapeutics of a disease, particularly chronic diseases, but also we are using it for telemedicine, remote medicine, use in hospitals to improve the management of patients. The whole idea of using this kind of wearables is, in fact, to try to do a personalized approach to the treatment of patients. And the idea here is to know how each person actually behaves in a way so we can treat specific symptoms and address all our management of the care, as well as obviously for therapeutic management and therapeutic trials. So we do have a levodopa meter that I'll be discussing, as well as a fall meter, a continuous blood pressure monitor, as well as ways of testing an early diagnosis by using alpha-synuclein in spinal fluid. I'll be discussing the first three if I have enough time to do so. The idea of having pilot studies is to be able to do novel studies that cannot be otherwise done without any pilot data, without any data that is presented to show the feasibility and the fact that this can be actually something that is worthwhile pursuing. So the studies with levodopa meter has allowed us to get funding from NIH, as well as those with early diagnosis. And in the others, we are actually trying to work on the development of those grants. So let me explain why we are interested in the progression and measurement of the levodopa. Let me explain first that here you have what we call the concentrations of the levodopa that unfortunately don't last too long in blood, only an hour and a half. And you can see here what is the peak dose of the levodopa. And we can see as well here what is the efficacy of the levodopa. That is the amount of medication that is needed in order to get to an on or having benefit from the drug. That is what we call the efficacy threshold or the benefit for the medication. And here we can see that in early stages, we don't get to the threshold for the dyskinesias that are truly what are leading the major, what we would consider toxic in some ways, although it's part of the evolution of the disease, but use of the levodopa. Therefore, what we have here is what we call the therapeutic window. And so initially we need very little medication in order to get to the on, and we don't even get to the dyskinesias. But as the disease progresses, we can see that that therapeutic window narrows. And so it is kind of easy to go from the off to the on. So we want to get to know the amount of levodopa because that is in blood, because we want to get the minimal amount of levodopa and get the best possible treatment at any stage of the illness. So in addition to the motor fluctuations, there are also non-motor fluctuations: anxiety, drenching sweat, slowness of thinking, fatigue, irritability. And some of you may know that what we do is to ask people to note when they are dyskinesias and when there are fluctuations, whether there is an off period where there are dyskinesias, if mild, severe. But this is tedious, and some people have difficulties. Therefore, there has been development of what we call the PKG, a watch that measures the amount of movement and compares it to the general population. And we can see whether there is dyskinesias, whether there are bradykinesia, or the patient is doing well. But that doesn't help us to understand what is the minimal amount of medication. Therefore, this is the role of the levodopameter. That is a needle, that is a microneedle that pretty much gets to the interstitial fluid and allows the measure of the amount of levodopa with assembly that Professor Wang will explain. Another project is to look at the falls. So what is it that leads us to have a fall is something that most people, when they get to the emergency room, do not know. And falls are very common in the elderly, but they are even more common in the people that have Parkinson's disease or Parkinsonian disorders. So we are developing a device that is a phone that has several ways of measuring the gait, balance, freezing, orthostatic hypotension, as well as the normal causes that could lead to falls, that are heart problems or seizures or other problems that may occur. We also collect information from each person. So here it is, the program. The persons can put their own symptoms, when they took medications, etc. And there are sensors that go directly by Bluetooth to the phone. And in the back of the phone, there is a way of measuring and analyzing all that. That gives us a graph that measures blood pressure, heart rate, and freezing, etc. Another project is the measure of blood pressure. And Professor Long Gardner and Shen Xu are looking at a patch that actually looks at a very novel way of looking at continuously at the blood pressure without using a cuff. And that is using ultrasound technology that allows us to detect the walls of the blood. We have studies that are open to enrollment in those particular causes. So Wearables are extremely important because they allow us to get objective measures of symptoms, progression, allows clinical management, therapeutic management, as well as help us as having an outcome measure in therapeutic trials. I cannot say, or I can say that without our group that is multidisciplinary, we wouldn't be able to provide the state-of-the-art care that we provide and doing all the research and outreach that our group does. I also want to thank Professor Wang as well as Professor Young, that are the ones that have the wearables that we are that I just described. I want to thank you all for listening to me, and I would like to let Professor Wang to present his slides. Professor Wang has been collaborating with me for the past 6 years, and we have been trying to work on all these different projects, and we have like 4 projects or 5 now, because he's extremely active and is a clear scholar and very enthusiastic, as you'll see. So go ahead.
Thank you, Irene. As I say, I'm coming from the engineering school, and we have a fantastic bioengineering Institute of Engineering and Medicine, and I'm the director of the UCTV Center on Wearable Sensor. This is an agile sensor in the School of Engineering, which is responsive to societal and industry need. And the goal is to develop a next generation of wearable sensor to address really big challenges and big questions, bringing together broad expertise from different departments, engineering as well as medical collaborators like Irene. And also we hope to be a resource, outreach resource, as well as for research development and economic outreach for the state and also for the nation. So Irene mentioned some of my colleagues. This is Professor Sheng, that she work on the blood pressure, TP Zeng on the fall. So we have 20 faculty. This is myself smiling with my co-director. We have medical people, all aspects of wearable, from device to data to material, and really complementary background to allow us to tackle these challenges. Now, you know, the field is already almost 2 decades from the digital revolution with all the smartphone. We see a lot of movement to watches, wrist device. Now, most of these devices, which are really changing and improving the quality of life, they're focusing on mobility and vital signs. So what we have currently on the iPhone or the watch, Apple Watch, you can measure your ECG or steps. But what we are trying to give information about chemical biomarkers that Irene mentioned, like levodopa or other biomarker, and to do so by bridging the gap between this biochemical analysis and the digital world, so we can have it on a smartphone, all this data, and measure it continuously at 24/7, anytime, any location. So we are in the center, we're developing platform not only for medical, this is like what we dream for levodopa, but this is a glucose meter from Dexcom here in San Diego for monitor fluctuation in glucose, lot of fitness and performance for athlete and for soldier, forensic application and entertainment, again, 24/7. And the advantage of this is that you can give continuous chemical information, again, anytime in any location, and do it without piercing your skin to get blood sample. These are non-invasive. You put it on the skin and you measure your sweat or tears, and hopefully this will change the way they monitor the Parkinson patient and also wellness in general, and eventually can be done remotely in telemedicine at lower cost, which is now accelerated by the COVID-19 pandemic, moving to remote and non-contact medicine. So what we are doing in our lab, my own lab, I focus on chemical biomarker and doing it not only in blood, which is more invasive. We want to have a non-invasive of measuring the sweat or under the skin, the ISF, or even measuring saliva or measuring tears. So we have different devices that can analyze all of this. This with the microneedle we'll talk later on, but different tattoo, different mouth guard, even on glasses. And by doing so, we can measure. If you visit my lab, Irene saw it before, you'll see this mannequin with many devices that do not compromise the analytical performance or the comfort and daily activity. You have device on glove for forensic, device in socks. So we have a smiley tattoo for pH of the sweat, or a ring for measuring mobility or nerve agent, again, a mouse guard or a device for ECG, and lactate for sepsis. And we work also on the blood pressure with Schenk. So translating this for Parkinson, we started with Irene about 5 years ago, developing disposable strip. And we do it a lot what we learn from diabetic, where you have strip for glucose. The same we develop with Irene, a strip that do frequent measurement of levodopa at a short time interval. You can take 5 microliter of blood and with enzyme that recognize the DOPA. This was early work published in 2014. More recently, we started the levodopa-meter, which now we have a joint NIH, where we now dreaming to have a part of a closed loop system eventually, like in diabetics. So a lot of this drive by the lesson that we learned from diabetic, and I've been doing it for 3 decades, working on glucose from the strip in the late 80s. This is the common, what you see in a CVS or Walgreens, that in 5 seconds measuring your blood glucose. To more recently, this is the Dexcom 6 here in San Diego that monitor the ISF glucose continuously. And this is the ultimate goal. This is a CGM for continuous glucose monitor, and we are dreaming with Irene to have a CDM for continuous DOPA monitoring. So this is what inspires us. Can we translate this, the lesson for glucose to Parkinson? One challenge that glucose concentration are quite high. Glucose in blood is what we call 5 millimolar, and levodopa in blood is 1,000 times lower. So this is part of the challenge where we need to push. But the glucose is very advanced in terms of the technology, but in the levodopa, we have to tackle mainly the lower concentration. So again, the dopameter, to do it in blood with the strip is easier, relatively easier compared to the continuous. So this is what we published with Irene 7 years ago. These are disposable, mass-producible, screen-printed strip, like one of them, and instead of glucose, we have enzyme tyrosinase that modifying this carbon electrode. And then when you get the blood, it recognizes the DOPA, and in 10 seconds, you can get a readout of the DOPA level in blood. So this is early work. But as I mentioned, our goal is, again, like in glucose, where we have artificial pancreas, we want to develop a closed loop system where you measure continuously. This is a work from Medtronic, that they have a closed loop artificial pancreas. So where a minimally invasive sensor will be processing the data and actuate autonomously, eventually the DOPA pump that Irene have. So here we have insulin pump, which is here, also from Medtronic. So this is a closed loop, autonomous feeding. We call it sense and act. So we sense on the sensor and act with the pump. And this will be done eventually with algorithm, with AI, machine learning, all autonomously. So this is the first example of such closed loop in personalized medicine, and we want to adapt this concept for the Parkinson patient. So again, we will rely on microneedle that are, and these are tiny devices, 1 millimeter deep, so eyes. So they are really called minimally invasive. It's even smaller than the needle in the glucose. The needle in the glucose are 8 millimeter. Our needle, you hardly feel it. And again, the goal is to have a closed loop that you sense the dopa and deliver, based, as Irene showed, the narrow therapeutic range. So there are already several commercial wearable sensors for monitoring motor, motility, accelerated gyro. These are commercial, but these are physical sensors. We would like to merge our chemical sensing with this physical sensor to get complete chemical information and also information about motor action of the Parkinson patient, both in preclinical and more advanced stage of the disease. So this is our vision, to have a complete combination of the minute dopammeter, the chemical sensor, with the motion sensor, all with our processor that will drive the drug delivery. So there are different platforms that we can use to do the Dopa sensing. You can implant a big catheter in the blood. This is more challenging because this is in the blood. We're also working in Irene to monitor Dopa in sweat, like we do for glucose. So we would like to have alternative, completely non-invasive. With the microneedle, which we are doing now, we're shown here, these are minimally invasive. Really, you don't feel it, and it's really negligibly invasive because it's less than 1 millimeter. But we're also dreaming to have a completely non-invasive, using a wristband or a watch, a tattoo type, that we can measure sweat Dopa. But we will focus on the meter, which is we pioneered with Irene 2 years ago. We have the first paper where we show in vitro characterization. And the beauty that we, on this needle, which is 8 by 8 millimeter, less than 1 centimeter square, we put multiple sensors which give different information for levodopa. One is the enzymatic and one non-enzymatic. So we can build on this microneedle. You can see here 3 microneedle. This is again 1 millimeter, go under the skin. And we, in addition to DOPA, which can be measured by different technique, we can also monitor other related parameters like glucose, which some of the Parkinson patients are also suffer from diabetics. So we can have a more comprehensive information, what we call multiplex detection, monitor several Parkinson biomarkers in really pain-free approach. So the system that we use, we already, during this NIH project, we developed a platform. It's a fully integrated platform, not only the microneedle, but also the supporting electronic and the battery. All of these are enclosed in this enclosure. This is my center of wearable sensor. So we have the microneedle facing the skin, connecting with the electronic control, which apply voltage, measure the current signal, powered by the battery, and you can also do wireless transmission and wireless charging of this. So this is look like this when it's on the body. This is from the top, as you can see, and this is the part at the bottom. And this part is disposable, reusable after 1 or 3 days. But the other part is reusable, the electronic with wireless communication, wireless charging, and the battery. All of these are reusable. So only the sensor, which may cost $1 or $2, you have multiple needles. For redundancy, we can measure levodopa on 8 different needles to make sure that it's redundant, accurate. We take the average. And again, it's all wireless transmitted to the smartphone. So these are examples of data where we published with Irene 2 years ago, where in the same sensor we can measure levodopa using the enzyme, which look like this, and this is non-enzymatic by direct scanning. So you have what we call a multimodality detection, kind of complete lab on the skin or under the skin, which by having 2 modality, we can have more reliable, more precise measurement for more precise dose regulation later on. So at the moment we are adapting, we developed the system integration, and in addition to the under the skin, we are working, I told you, on levodopa sweat dopammeter, where we take the sweat from the fingertip and measure it at 10 minutes interval without taking any blood. So this approach is the one that will be the dopammeter with the NIH for continuous measurement at a 1 minute interval. So there is fast fluctuation. And in this case, we can capture dynamic changes and respond accordingly using this dopa meter needle. And this is, again, this is a relatively fast 10 minutes. And the beauty is all in sweat. We don't need to go under the skin. And our preliminary results are very encouraging. This is where we take the dopa pill, and you see every 10 minutes, you see the kinetic profile. This is data, is the raw data. After 20 minutes, increasing 30, and then it's decreasing again. So this is another exciting project, non-invasive. So overall, we have a large group of very dedicated postdoc and student and visitor from a wide range of expertise, from material, the chemical. In this specific project, we work with Azir and Farshad, who work on the microneedle, but they all supported. Here we have Rodolfo is also working with Irene for drug delivery. So it's very exciting. This before the pandemic, we meeting just last year. And again, support of the Center Wearable, Center Engineering and Medicine, and joint grant, NIH grant with Irene, fantastic collaboration with Irene and Katie. So we hope together to shape the future of Parkinson care and Parkinson management. Thank you very much.
So exciting, Irene and Joe. This is really what makes it so inspiring to be at UC San Diego, where there's cross-fertilization across departments like neurosciences and engineering, the Center for Wearable Technologies and Nanoengineering. And I always marvel at how much in medicine can be attacked from different perspectives. And I think, physicians spend so much of their time studying the biology, and they may have ideas of, wow, wouldn't it be nice if we could do this or that, but we don't yet have the skills or the time to understand things like nanoengineering. And so having collaborations with folks like Joe Wang in the Department of Nanoengineering, where one of the first in the nation actually established through Joe's work, really makes UC San Diego a truly special place. So thank you so much, Joe and Irene. And right now we're going to be able to open it up to Q&A, which is very exciting. So you in the audience will have direct access to these world leaders in Parkinson's and nanoengineering. The first I'd like to put forward to Dr. Wang from an anonymous attendee. Would the sensors have to be attached to you at all times? How would you manage that?
No, yeah, if you want continuous readout, you need to contact under the skin to expose. So yeah, it's exposed, but it's really negligibly invasive. It's less than 1 millimeter long. So when you remove it, there is no sign, nothing, no irritation, no skin effect. So it's really negligibly. And the company are already exploring it for diabetic with FDA approval.
Yeah, that's what I was wondering, because, being here in San Diego where Dexcom is, and really understanding what are the challenges. Certainly you noted the challenge of, DOPA levels being 1,000 times lower. Do you see that as a major hurdle to getting toward FDA clearance and the ability to widely apply this?
That's a good point, because the other challenges like biofouling or passivation has been addressed in the glucose. So people, and I know how to address this protecting the sensor from passivation in the under the skin, but the concentration is what we are trying. We have in vitro, we demonstrate we can go to lower, below the, below micromolar. We can go to this level, but still on the body, as we just recently integrated the system, we started this, and we're testing it with pills, and we see preliminary results. Surprisingly, the sweat is even more responsive. So we have very surprising results using sweat, but the sweat is not continuous. It's every 5-10 minute interval, which is also exciting, replacing the blood with sweat.
That's amazing. That's incredible.
A
A question for Irene. When you look at this technology and the amazing capacity of this work, how would we apply this to other, what's called, at least one of the anonymous attendees is saying, how would it be applied to all PD types? And do you see a way that this could be applied beyond just standard Parkinson's disease, or do you feel like there may be a means to apply such technologies to things like, maybe multi-system atrophy or other sub types of movement disorders?
Yes, this is a technology that can be used for everybody, particularly those that have to do with, for example, measuring whether someone falls. That is a perfect example of how this can be used. For example, patients that have multiple system atrophy have very low blood pressures, and they may have a syncopal event when they stand up. So this fall meter, for example, could be used for that, to let the person know, hey, your blood pressure is too low, don't get up at this point. So that's kind of where we're trying to go with this discovery. So go to a second stage, that is using the information not only to learn that something is happening, but also to search for treatments. Regarding the levodopa meter, because levodopa in general doesn't work in some of the typical Parkinsonian disorders, for those that would not be useful. But for multiple system atrophy or dementia with Lewy bodies, certainly that is a technology that can certainly be used, because we want to use the minimal amount to produce the least amount of side effects. And in these two disorders, for example, there is low blood pressure. So, and the levodopa by itself lowers the blood pressure. So we want to use the minimal amount of levodopa to be able to treat, but not produce side effects.
Great. Thank you so much. Here's a question regarding, what do we think these sensors will end up costing? I know that's difficult to say at this time, but do you feel like these will be affordable technologies, Dr. Wang?
Yeah, electrochemistry by itself is a low-cost technique. So if you look at the glucose strip, people can buy a monthly supply for $50, and the meter, they get it for free. You get, so the disposable, like razor and blade, that the disposable are the strip. There's still a $10 billion industry. The diabetic market is over $10 billion. It's
Wow.
we have 28, 30 million compared to Parkinson. But even the microneedle will be probably eventually $20. But I told you also we have disposable. The disposable part will be only $1 or $2. The reusable one you can buy for $20 and keep reusing the electronic portion. Yeah.
Very exciting. And I presume that non-tech savvy people will be able to use these sensors. I don't believe you'll need necessarily a lot of technology.
Oh, yeah, we make it as user-friendly. Even grandma can do it by herself. Yeah, that's what they do in diabetics. They do diabetic. In 10 seconds, you get the blood glucose. Amazing, these grandmas.
Great. Question about the wearable sensors. How might you be able to disperse the medication? And this may be for Irene, so the level of therapeutic dopamine will remain constant. It seems like that's one of the sort of holy grail of being able to, when the narrow therapeutic window starts to happen later in the disease, and it's very difficult to hit the sweet spot of the dosing, and how one might be able to use wearable sensors to address that question.
Well, that is exactly where we want to go. And I think that will be a paradigm shift, as it was the use of the glucose meters. The idea is to try to see, okay, for this person, this is the perfect level. And we get to this perfect level by this amount of medication. And it can be oral, it can be any other way that the medication is given. So yes, this would be an important way to understand that. And I think it is also important because some people do have problems that are the non-motor ones. And those are so much more difficult to nowadays understand what is the proper level. And so this would allow us to know, okay, the proper level for anxiety is this one versus the motor one is this one. So this would be the proper level that we need to use. And this will be the certain amount of medication that needs to be given. All that needs to be studied in detail, and that will be, taking time until we know what is the proper level for each person. But I think that should not be very difficult at all. As we know what happens with glucose, right? We know the levels, and then we give the certain amount of insulin, for example, or oral medication for that matter.
Great. A brief question for Dr. Wang. What is keeping the levodopa sensor attached to the skin besides the needles? Are you using some sort of special adhesive to?
Yeah, the commercial adhesive from 3M, they're just keeping very user-friendly, user-independent. So it's very easy to remove at the end of the day. So it's everything is user-friendly. When you remove it, you don't see any memory, no irritation, etc.
Great. So one attendee notes, as I understand correctly, if I understand correctly, most of these are being developed right now. What do you need to reach completion? How soon do you project some of them to be available and used as designed?
So again, a lot of this much easier if you go to the sport, the fitness, performance market, then you don't need the FDA approval, especially when you involve drug that is response to the meter. So anything involved, both there are two aspects. One is still going under the skin. So we got IRB, we don't have any issue with microneedle, but anything that involved later on make a decision based on the sensor to dose the levodopa, FDA will really need to, this will be the bottleneck eventually, yeah.
Right. And what other, can you expand upon a couple of a combination question here? One is, are there any other devices like this in development? And then the other is, which sensor do you feel is most accurate, and are the sensors presently being used in clinical medicine? So basically talking about broader applications and other sensors that might be out there that may have some alternative benefits and drawbacks.
So in general, as I say, always glucose is leading the market, but there are a lot of activity for lactate, for sepsis, cortisol, for stress and performance. The military is interested in cortisol. And again, we do a lot of lactate, alcohol sensing. We work with Sandra Brown is expert on alcohol. So all type drug of abuse, opioids monitoring for kids, all type of interesting. And then also security. We were with the Army for detecting nerve agent, all type of warning the soldier, protecting the soldier for explosive or nerve agent attack, etc. So amazing. One example, the last one is with the Navy here in San Diego. We do underwater explosive. We put on a wetsuit of a diver or wetsuit of a surfer to put this sensor on textile, or you can dive and look at explosive residue. The Navy want to protect the Navy boat. So we work in Point Loma with the Navy people to do this underwater sensing of explosive residue. This is
I guess to further explain on the specifics of detecting DOPA, it sounded like you felt very optimistic on the sweat approach. The question of whether you think which is most likely to win the race here, the sweat approach, the blood approach, other approaches?
No, okay. And even in the case of glucose, dreaming to replace the needle, the CGM, go completely non-invasive. Remember, Google have a smart glasses. So Google tried to have glass for tears glucose. So we are keep dreaming, tears, saliva, sweat, ISF, But again, the ISF under the skin is closer to the blood, is well established. So that's why it may be winning the race. But the sweat has become very interesting, and also for sweat glucose, etc.
Let
Excellent.
me add also that none of this technology is being developed anywhere else. We have asked many our excellent scholars throughout the world, and they all supported the idea, but nobody has been developed any of these devices. So this is really very, very novel.
Wonderful, Irene. I love that segue into talking about our special location here. I really think even centers, very well known centers that have innovated a lot in the care of patients, they don't have the special sauce that UCSD has, where this incredible center is in a place where you can have an interaction with, walk across the bridge here and interact. And now with Zoom, I guess it works quite well, but it's just remarkable, this place. It's been described as a playground for neurosciences. I think, because we really have just such a robust environment, intellectual environment, and no real silos to really halt, interactions and saying, oh, that's my project, I don't want you getting involved in it.
Right.
The special entrepreneurial spirit of UC San Diego, having been here actually really quite young as a university in terms of where it is. I mean, being #1 in the nation in NIH funding in neurosciences, and yet only being 60 years old or so, I think really describes our special environment where, maybe some of the older staid institutions have built up too many silos to be able to have that kind of cross-fertilization. One last question for you, Irene. When you look at this really promising new collaboration and thinking of the treatment using these tools, could you one question here is, would patients start to use them for diagnosis or later in the disease development? I don't want to lead you, but I think it is quite exciting, maybe on all ends of the disease, where one might be able to be very creative in using this in the diagnostic process. But can you expand on that?
Yes, the idea here about diagnosis is once we develop ways in which we can measure a specific protein in the blood, and we can identify it as a way to diagnose a disease, for example, abnormal alpha-synuclein in blood, then we can use these sensors to measure that. And so that would help with diagnosis. So as soon as symptoms or family members or whatever would occur, or even exposure to pesticides, for example, one could diagnose early on this disease. So that is extremely, extremely exciting. Of course, later on is very clear because we can use it not only for diagnosis, but also for the management in many ways, as we describe, right? I mean, the way of measuring how we walk, the measure how we have symptoms of anxiety or whatever else that could occur can be measured, as well as, of course, the levodopa.
Great. Well, I think, and as so one thing I didn't mention to the attendees is that I direct the Charlie Marcos Alzheimer's Disease Research Center, where we have great collaborations, and Irene is part of this program. She's really highlighted the benefits of being able to access fluids for biomarker analysis. We now have plasma biomarkers for the main proteins of Alzheimer's disease that are looking quite promising. And I think this work is going to influence the kind of biomarker measurements in Parkinson's disease as well. I think we'll call this the final question from Alan Kleiman. Can the delivery of DOPA be adjusted to a temporary need to control dyskinesia, for example, prior to driving a car? I think that's a really exciting idea to think of, oh, I need to dial it up just a little bit. I see another thing that in our neurology department, we cover such a breadth of diseases in neurology, you see this in things like myasthenia gravis, where a person gets weak at a certain time and might need to say, well, I want to try to chew a steak, so I'm going to take a little extra of that medication so that I get a little more strength, but I don't need that strength at all times. Could you do such a thing in Parkinson's disease?
Absolutely, absolutely. And so we have people that do take extra dose prior to playing golf, for example, and this would be the same.
Wonderful. Well, thank you all for a very enlightening conversation. I think this is a great kickoff to the week. I'm so excited to be with you all week, and hope you all will come back tomorrow for at noon for Care Beyond the Clinic, a community support programs. I'll be here to introduce it, and we look forward to hearing your questions. And thank you once again for your time with us.